All right, couple quick announcements. Again, exam number 5 due on Friday. Obviously, if you want to turn it in earlier, you're welcome to if you think you've had enough time on it. With that, it is we don't have an official class on Friday, but I will be here if you guys want to have a study session during the normal class period for kind of the final exam. The way that works, just bring questions that you might want me to talk about or cover again for that one. So Friday, it's optional. You don't have to come, but I will be here if you guys want to use it as a study session. If you're not going to be around on Friday to turn in the exam, just turn it around just about, I should be around just about all day that day if you want to turn stuff in. I'm still working on the unknowns project. That's due Friday then as well. And that's the gram stains, the streak plates, and then the unknowns charts, which are up on those turned in electronically to a Dropbox on Brightspace. Okay, questions about any of that? And then final exams on Wednesday, 3:30. So a week from today. Not that far off. Okay. All right. Today, though, we're going to finish up kind of talking about the last little bit of material on viruses. So I left off kind of talking about lysogenic versus lytic viruses. Today, I wanted to also talk about a few other kinds of viruses. Specifically, I wanted to spend just a little bit of time reviewing what we talked about yesterday, but in the light of human or animal viruses. There's many diseases like colds, flu, cold sores, chickenpox, COVID-19. They're caused by viruses. And there is some common themes, though, when it comes to human/animal viruses, though. So with these, most viruses that infect animals are actually enveloped. Not all of them, but many of them are enveloped. Animal viruses in particular tend to be enveloped. And this is because the envelope will help with both entry and egress, exiting of the viruses. And that viral envelope really helps with both of those processes, particularly with animal cells. And it's partially just because animal cells do not have cell walls. So because animal cells don't have cell walls, this actually makes having an envelope really help viruses both enter the cell and exit the cell. So in the case of animal viruses, again, they tend to be enveloped. Not always, but a lot of them are enveloped. And then they have those spike proteins that come out. So these are our spike proteins. Again, spike proteins are involved in host cell attachment. So with these animal viruses, they'll attach via those spike glycoproteins. And again, remember, we talked about this last lecture, but they bind to specific proteins or glycoproteins on a host cell surface. So there's going to be some type of specific protein on the host cell surface that they recognize, and those spike proteins then bind to. And that triggers entry into the host cell. Typically, when they enter into the host cell, when it comes to viruses that infect animal cells, they usually enter in by either fusion or receptor-mediated endocytosis. And we talked about a little bit, very briefly, on both of these processes last lecture then as well. But if it is entering in by, for instance, fusion, again, this is just going to be the viral envelope. You can kind of see all of this right here is the viral envelope. The orange is the capsid proteins. Maybe the blue is the then DNA or the RNA, the nucleic acid genome of then the virus. But what ends up happening is literally the membrane envelope of the virus fuses with the membrane of the host, and that allows the penetration of the virus into the host. So the membrane, if this is the envelope, it hits the plasma membrane, but it fuses with it in such a way that it spills the virus inside of the host cell. It might also be, though, that it triggers receptor-mediated endocytosis. So again, this might be the viral particle coming in, but there might be specific receptors, proteins that the cell uses, and maybe those things are used to bring in food particles by endocytosis or phagocytosis, for instance. But in this case, though, the virus tricks the cell, activates these receptors, and it gets brought into the cell by that endocytic event. So again, it's usually entering by fusion or receptor-mediated cytosis, but the membrane, the envelope of the virus can really help with this, especially in the case of fusion. So it helps with entry. And then remember also those viral envelopes, we talked about this last lecture, they're derived from host cell membranes during the egress process. So a lot of times you have viral proteins that will insert into the host cell membrane during the synthesis of those viral proteins. You'll get viral matrix proteins that come onto here as well. For instance, I showed you a little bit of a video with that. But then this, when a virus particle docks with this area of the membrane, it triggers budding, where the envelope is made out of just that host membrane portion. This is just a consequence of how the virus exits or egresses out of the cell. But this is how it then gets out. And a lot of times these buds come off over time. And the viruses can actually bud off continually over time as well. But that having that envelope helps with both the entry, especially by fusion, and then the envelope is actually a consequence of how it exits the cell, the egress process then as well. Questions about that? When it comes to animal viruses as well, sometimes you can have what are called acute infections, latent infections, persistent infections. An acute infection is a strong but short-lasting. Infection, and you can actually see this graph here kind of goes through the difference of what is an acute versus persistent versus a latent infection. An acute infection is where you have a large number of viral particles in the body, but for a fairly short period of time, usually about a days. So for instance, it's something like flu would cause an acute infection. However, what general type of virus would probably most commonly be causing more acute infections? Acute infections, the virus is in there for a short period of time and then goes away. But they're usually also talking about these being more lytic / virulent viruses, or no, not always. Latent infections is basically usually when we're talking about latent viruses, you have an acute stage, but then later on, you have a time period where there's no virus in the body, or at least no detectable virus in the body. And then sometime later on, you have that virus in the body again. Latent infections is where the virus goes dormant for a while. What kind of viruses would be causing more latent infections? Chickenpox would be an example of one that causes latent, it can have a latency stage to it.
Oh, lysogenic.
Yeah, lysogenic. And what's the term for lysogenic viruses? Not virulent viruses, but the other one, temperate. So when I say latent, I mean usually there's an initial infection of the virus, and then the virus is still in the host cell, but at no detectable levels. And then at some later time point, it can wake back up and start reproducing again. This almost always means this is a virus that followed the lysogenic cycle, and it was hiding inside of host cells in the form of a provirus, the viral DNA inserted into the host cell chromosome. So chickenpox does that. HIV does this, for instance, then as well. Persistent infections a little bit more unusual, but they're bad. Persistent infections, this is where it just goes on for months to years. as a constant level of viruses in the body. This is often only typically happens in individuals that are immunocompromised, for instance. But you might also notice that sooner or later, it just kind of my blue line ended. Persistent infections are bad. It usually ends up being sooner or later the host self dies from those because they can't fight them off. Okay. That there's usually some complicating factors involved in those, though. All right, the last grouping of viruses I want to talk about is RNA viruses. RNA viruses are of particular concern when it comes to human disease. RNA viruses, why are they called RNA viruses? They have a RNA-based genome. The thing about RNA is less stable than DNA. RNA is just naturally a less stable molecule than DNA is. What do you think that might mean about RNA over time if it's less stable? Changes over time or mutates over time. What that means is if RNA is less stable than DNA, what that also means is it mutates, changes more quickly. And since it mutates more quickly, RNA viruses then also change more rapidly as a result of that. DNA viruses have pretty slow rates of change because their DNA is very stable. RNA viruses can change much more rapidly. Why would that be a problem if we're talking about disease then?
Hard to like keep up with like vaccines.
So it makes vaccines less effective because the vaccine is going to protect against the version of a RNA virus from last year. Maybe the new version of the RNA virus this year has changed enough so the vaccine doesn't work anymore. What is really just so happening, though, also as it changes, it looks different to our immune system. And what that means is you can lose secondary responses. And if you're losing secondary or memory responses, that also means vaccines are not as effective because what do vaccines do? They give you an adaptive immune response, a memory response, without having to get sick from the disease. So what you get, do you guys know what's in the vaccine shots when you get a vaccine? It's usually, sometimes it's the actual virus, but maybe a dead or a weakened version of it. Sometimes it's just the antigenic piece of the virus or the pathogen. But basically, when you get a vaccine shot, what it does is just parts of a disease. Maybe a whole part, maybe just parts, though. And what it does is your immune response then develops a memory response against it without ever having to get sick from the disease in the first place. So you basically get a secondary memory response, and that protects you against getting sick from it. So anytime I say you lose secondary responses, that means actually you're losing your memory responses so you can get sick from the disease again, but it also means vaccines don't work as well because that's how they work. Questions about that? So yeah, you lose secondary memory responses. It lowers vaccine effectiveness, which is related. Okay. So there are some RNA viruses, including influenza viruses, HIV, Ebola, SARS-CoV-2, which is the virus that causes COVID-19. By the way, this stands for Severe Acute Respiratory Syndrome Coronavirus number 2. It's a very technical name. You guys will. Anyways, and what you might also notice that most of the diseases don't really necessarily have great vaccines. Like we don't have a vaccination for HIV. Ebola, we technically kind of have a vaccine for it, but it doesn't work very well. And then there's other ones like, oh, we have SARS-CoV-2. Do we have a vaccine against it? Yeah, but the vaccines they give out now are not the same ones they were giving out when they first came out 2 years ago. Okay, the vaccines they're giving out now are adjusted, and they are different ones. They're actually adjusted to target current variants in circulation. The same thing with flu viruses. Do we have flu shots? Do we have flu vaccines? Yes, but you have to get a new one every year because the flu has changed every year, and the previous version of the flu vaccination doesn't work against the new varieties or the mutated varieties of the flu that are circulating around now. Same thing with coronavirus. They're getting variants often enough that you need updated versions of the shot for it to have full effectiveness. Questions about that? Yeah.
When we get the flu virus or the flu shot every year, is it still effective? Like in 5 years, if I would get that strain that was 5 years ago?
No, probably not. Just, but it's less about the. Probably not. It's less about. It's just usually the flu changes at random, so it's never going to be the exact same variety. Does that make sense? Yeah.
Okay, so since like wild animals aren't vaccinated and all that jazz, like why aren't like animals dying like all the time? You know?
So a couple different things. One, they are, depending upon the disease. Two, flu for birds is like a cold. Flu is originally a bird virus. All influenza strains are originally birds, but they've been in bird populations for so long now that the birds have kind of evolved so that that disease is less severe for them over time. Okay, so flu in birds is like getting a cold, doesn't usually kill them. Flu in us is just more severe, for instance, because it hasn't, yeah. There's some different reasonings for that. There's a lot of context involved, but there are plenty of diseases like, for instance, Ebola has wiped out gorilla populations in Africa because it more recently hopped into those gorilla populations. So yeah, you have some of the times it does happen even in the absence of vaccinations that you do have disease wipe out populations. When it comes to RNA viruses, replication of RNA-based genomes, well, we are particularly concerned about RNA viruses, but there's some challenges from the virus side of even having an RNA-based genome in the first place. First off, do cellular organisms ever make copies of RNA off of other copies of RNA? Have you guys ever heard like RNA being used as a template to make more RNA? No. And so with these RNA-based viruses, they have an RNA genome, but how do you copy an RNA genome into another RNA genome? How do you copy RNA into RNA? Cells can't do that. So cells like our cells cannot make RNA off of other RNA. They don't have any of the enzymes to do that. So what that means is the virus has to have the enzyme to do this. So what I'm really going to hit here is how do RNA viruses copy their RNA genome? So we're going to look at a life cycle of an RNA virus real quick here. So if you have an RNA virus, it's got an RNA genome. It enters into the host cell, and then that viral genome encodes a gene that makes a protein called RNA-dependent RNA polymerase. It's a little bit of a mouthful, but it's an enzyme that polymerizes RNA dependent upon an RNA template. Again, you read them backwards, you get what they're doing. Okay, so some viruses, what they do is they use RNA-dependent RNA polymerase enzyme to replicate RNA into even more copies of RNA. So they're using their RNA as a template to make copies of that RNA molecule. So again, with these RNA viruses, what they're going to do is they enter into the host cell. When they do that, though, they release their viral genome, their RNA genome into the host cell, and that's what I have in red here, for instance. RNA polymerase, RNA-dependent RNA polymerase, this enzyme complex is going to bind to the RNA, and it copies that into a messenger RNA copy. And then sometimes also it copies that into a genome copy then as well. So you have to copy the first strand into a RNA molecule, but then those RNA molecules can get used as messenger RNA and making viral proteins, or that messenger RNA can also be used as a template to copy the entire genome of the virus itself. Questions about that? So RNA viruses, they have this RNA-dependent RNA polymerase enzyme. Okay. That being said, there are actually what are just called RNA viruses, and then a different grouping of viruses that also use RNA as a genome. So there is just the kind of RNA virus, and what we refer to as RNA virus is just that. But there are other types of viruses that fall into what is called a retrovirus category. And retroviruses, they do have an RNA genome, but their life cycle is more complicated. And retroviruses are specifically going to be types of viruses that have an RNA genome, but at some point in their life cycle, their replication cycle, their genome is also going to enter into a DNA intermediate stage. Okay. Again, retroviruses have an RNA genome, but they're not, I'm giving them a separate grouping from what I just referred to as RNA viruses. All right, this makes a little bit more sense if we go through this a bit more step by step. And the kind of classical example of a retrovirus is HIV. Okay. Animal viruses that have the most complicated life cycle are these retroviruses. And retroviruses, they have a special enzyme called reverse transcriptase. I'm going to get to that function here in a second, but all retroviruses use an enzyme called reverse transcriptase. And again, one example, and I'm going to use this for going through retrovirus replication, but one example of a retrovirus is HIV, human immunodeficiency virus, and this causes AIDS, which is the disease, acquired immunodeficiency syndrome. All right, so if we're looking at HIV and how it affects host cell, HIV is an envelope virus. It's a retrovirus. Off of that, then these envelopes, it has the spike proteins. So here's my spike proteins. The spike proteins allow the HIV to bind onto specific protein receptors on the surface of very specific white blood cells. Specifically, they typically bind to things like T helper cells, although a couple other kind of varieties. So this my big host cell is supposed to be a T helper cell. Somewhere on this cell surface, it usually has what are called CD4 proteins on its surface. And CD4 proteins are essential to the function of a T helper cell in our immune responses. So they have to be there. They're part of the T helper cells and how those T helper cells get activated. But what the HIV does, again, my HIV particle come in here, and the spike protein of an HIV will bind onto those CD4 proteins, and then that triggers entry into the host cell. And so then it affects cells of your immune system, specifically it affects T helper cells. It's been a couple lectures and I went through it really, really quick. Do you guys remember what the main function of T helper cells is? Close. You're getting T helper cells mixed up with the other one, which was T cytotoxic cells a little bit. So that's more the function of a T cytotoxic cell. Or a phagocyte, maybe, is what we almost might pick up, but these aren't going to be phagocytes either. T helper cells are the major cytokine producers of our immune system. And what really just happens is HIV gets inside of T helper cells and then makes it so they don't function properly. Remember, HIV causes acquired immunodeficiency syndrome. Immunodeficiency syndrome just means the immune system is deficient, is not functioning properly. Why is it not functioning properly? Because HIV targets T helper cells, shuts down their function, and then you can't make enough cytokines to activate robust immune system responses. So people with HIV have depressed immune systems or deficient immune systems. It's because HIV is specifically targeting this cell type. Questions about that? So it enters in typically by fusion. So the envelope is going to fuse with the host cell membrane. I'm just kind of showing this in some electron micrograph images over here on the left. Once inside, the capsid proteins are removed, and then that will cause the RNA genome to get into the cytoplasm of the host cell. However, inside of that viral capsid is not just the RNA genome, but a few different enzymes, including then the reverse transcriptase enzyme. So here you can see in red is the RNA genome getting inside the cell, but again, it's also releasing in the green these reverse transcriptase enzymes in there as well. Reverse transcriptase, does the reverse of transcription. What does transcription do? Just normal transcription. Yeah, it uses DNA as a template to make RNA. What would be the reverse of transcription then? Yeah, and that's what it is uses instead RNA as a template to put together a DNA molecule version. And reverse transcriptase is the only enzyme that's capable of using this. It starts with RNA as a template and makes a DNA copy of it. Questions about reverse transcriptase then? That newly synthesized DNA will then integrate into the host cell chromosomes as a provirus. So it'll migrate to the nucleus, insert into the host cell DNA as that provirus. And then it can stay hidden inside of those host cells for a long period of time. Again, that would be a dormancy stage or a latency period. This is why people with HIV can go long periods of time, sometimes years and decades, with no noticeable symptoms in some cases. And many types of retroviruses do have these latency periods. The provirus actually never leaves the cell. So once it's in there, actually never leaves in the case of a retrovirus. Instead, what happens is it's copied into a RNA copy. So RNA copies are made off of that provirus sequence. And those RNA copies then act as both messenger RNAs to make viral proteins, but also as RNA genomes for the next viral generation for new virions. So some of it gets a messenger RNA copy gets made off the provirus. Some of that will get used to make viral proteins in transcription. Some of that will just become genome for new virion particles and just being used to make those new viral particles. Questions about that? And then the retrovirus typically doesn't lyse out of the host cell, so it doesn't cause lysis of the host cell. Instead, the virus will just egress by budding over time. So you have continual production of the virus over time. and it will bud off. And in the process of budding off, then it gets wrapped up in that envelope, which is made out of the host cell membrane. Question then about retroviruses. So, by the way, you don't, viral species, depends on if it's an RNA virus or a retrovirus, they don't use RNA-dependent RNA polymerase and reverse transcriptase at the same time. Retroviruses will use reverse transcriptase. RNA viruses will use RNA-dependent RNA polymerase. There's no overlap between them. That being said, why should we care about RNA-dependent RNA polymerase or reverse transcriptase enzymes? A little bit, actually. These two enzymes do tend to be sometimes a little bit more error prone. So it can cause higher rates of mutation in some cases. But yeah, so that could be part of it. Do you guys remember when I talked about antibiotics? They're useful because they're selectively toxic. What is selectively, to be something selectively toxic, what was the important characteristics of it? So it has to hit, let's go to that first point. It has to inhibit the growth of the bacteria. Antibiotics have to target something that's important for the bacteria. Are these enzymes important for the viruses? Do you have to have them for the virus to reproduce? Yes, in the case of these RNA viruses. Okay. What was the other part, Riley? Or did you get the second? It has to stop the growth of one thing, but permit the growth of others. How can it stop the growth of one thing, antibiotics, but permit the growth of, say, human cells? So it's got to target something important to the bacteria. Antibiotics do, but why does it then not stop human cell growth? So it targets something that humans don't have. So antibiotics target something that's important to the bacteria that it has, but that human cells don't have a version of, or that they have a substantially different version of. Do human cells make a equivalent of either RNA-dependent RNA polymerase or reverse transcriptase? No. These are virus-specific proteins. RNA virus make RNA and make and use RNA-dependent RNA polymerase. It's essential for that, but humans don't have this enzyme. Reverse transcriptase, essential to the life cycle of a retrovirus, but humans don't have it. So what I'm basically just saying is these two enzymes can be targeted. And if we can have a drug that specifically targets RNA-dependent RNA polymerase or reverse transcriptase, you can stop viral reproduction without harming human cells. And there are actually several drugs that target these two specific enzymes to then slow the growth of viruses or hopefully even maybe cure viruses, for instance. So most of the things that HIV positive patients take is they actually take things that target reverse transcriptase in some way. It usually never fully gets rid of the virus in their body, but it keeps the virus at very, very, very low levels in their body so that they don't have active symptoms. It just can't get rid of it fully because it's dormant and hiding inside of our chromosomes. So by targeting the reverse transcriptase, you stop the viral reproduction, but you can never get rid of it because it's in the DNA. Questions about that? By the way, there's several different drugs that target reverse transcriptase, but just to give you an example of a grouping of one, there's what is called nucleotide analogs. They look like A, T, C, or G. nucleotide analogs look like the nucleotides, adenosine, thymine, cytosine, guanine, for instance. And the reverse transcriptase does mistakenly use these nucleotide analogs, but when they use a analog, it actually creates a termination of the reverse transcriptase process. They're basically versions of nucleotides that don't have something attached, that 3' hydroxyl. So you can't go past that point. It stops the replication process after that. The thing is with these nucleotide analogs, they're designed in such a way that reverse transcriptase still tries to use them, but our normal polymerases like DNA polymerase or RNA polymerase inside of our cells don't use these things. Does that make sense, everybody? That's a little bit more complicated. Okay. All right, questions about viruses then overall, some of these broad groupings of viruses and what's going on with them. All right, as small as viruses are, there are some things that are even smaller infectious particles that do exist. So for instance, viroids are, all they are is circular RNA molecules. So it's kind of this, I know this doesn't look like a circle, but this is an electron micrograph of a viroid, but it actually has these stem loops that come around, but it is a overall circular RNA molecule that is just a loop of RNA overall. So viroids are circular RNA molecules, and they can cause disease in plants. They really only cause disease in plants, though. These small RNA molecules can replicate inside of plant host cells, and then they cause errors in regulatory systems that control the plant growth. So for instance, this is a normal, healthy potato plant versus one with a mild viroid infection, an intermediate viroid infection, and then a severe viroid infection in this particular image. It stunts the growth when it has these viroid infections. This is chrysanthemum leaf that has a viroid infection as well, where it's causing some lesions in the leaf then too. So viroids are just these small circular RNA molecules, but they only cause disease in plants, and that's it. Prions are another thing that is even smaller than a virus that can mediate disease. Prions are slow-acting infectious proteins. Very slow-acting, and typically they only are associated with causing disease in the brains of mammals. Some examples of prion disease is scrapie in sheep. Mad cow disease, you guys might have heard of that before. That's a prion-based disease that affects cows, but also can transfer from cows to humans and has done so in the past. Creutzfeldt-Jakob disease in humans, and then Kuru in humans. These are all diseases caused by prions. And what they do is they cause brain tissue to shrink and deteriorate over time. So these kind of blank open spots, this is some histological slides of brain tissue, should not have these open plaques. This basically is missing neurons in the brain that have been destroyed by the prion disease, and you get brain shrinkage deterioration over time. That then corresponds to things like loss of cognitive function, confusion, memory problems, failure to form memories, can even lead to then loss of muscle control and shakes over time, for instance, then as well. Okay. Mad cow disease is in cows, but you can get it from eating contaminated beef, for instance, but you would never know it because it takes about 20 to 30 years to set in after that, for instance. Very slow acting. Kuru is kind of interesting. This is actually a prion-based disease that was only detected in certain indigenous native tribes in like the Indonesia area, but they actually got it because part of their funeral practices was actually eating the brains of their ancestors. So somebody died, and then they would eat their brains to pass on their knowledge to the next generation. Unfortunately, that's actually one of the best ways to also pass on prion-based diseases. Prions, what happens is prions propagate by, it's an abnormally folded version of the protein. So if you have a prion version of a protein, it binds to the normal shaped version of the protein and causes it to misfold into the prion shape. And then those bind to more normal proteins, and then they convert them into the prion shape of the protein. And it's kind of like a self-propagating feedback loop. The prion, misfolded prion binds a normal protein, converts it into a prion, which then binds to more normal proteins and converts them into prions. And they don't have the right function. Again, there's just these misfolded versions of a protein, but that misfolded version of a protein propagates itself. So they interact with those normally folded proteins and cause it to fold abnormally as well. And that, again, just causes a slow feedback loop. But those misfolded prions will start to aggregate inside of the host cells. And as they aggregate inside the host cells, they start to interfere with the normal cellular function and eventually end up resulting in death of the host cell. However, it's very slow. And so it really only impacts types of cells that do not divide very often. So if you've got a skin cell, every time the skin cell divides, it takes these aggregates and dilutes them out into the two host cells, into the two daughter cells, for instance. But because the skin cells divide pretty regularly, it's always diluting back the prion proteins. So it never really causes an impact in, say, something like a skin cell. Does that make sense, everybody? How do nerve cells divide very often? Actually, hardly ever in human adults. So the idea here is, again, these aggregates build up really, really slowly over time. But because nerve cells never divide, the aggregates never get diluted out. And therefore, they build up over time inside of nerve cells until they cause issues in the brain and neurons. Questions about that? Prions are real tricky because cooking doesn't, I went back to this point, I said nearly indestructible. Things like cooking actually doesn't destroy prion proteins. The misfolded prion shape is very, very, very stable and propagates over time. This is a very, very rare event, but we have it kind of detected then as well. This is also why people who ate beef in the United Kingdom in the 70s and 80s are not allowed to give blood. You guys never probably heard of that because it probably has not come up in a long time. But it turns out that there was mad cow disease in beef populations for the United Kingdom during the 1970s and 1980s. And potentially anyone who ate that beef could be carrying the prions and not even know it, because they might have no noticeable symptoms. So they've just banned anybody that was in those locations and might have eaten the contaminated beef from giving blood, because they could have the prions in their bloodstream and could pass them on to somebody else and slowly mediate disease over time as well. Okay. There is a prion-based disease that's bad in Iowa at the moment. Anybody know any hunters? In Iowa deer populations, there's a disease that's going around called chronic wasting disease, and it's a prion-based disease. As far as I know, they cannot transfer into humans, but I'm not in a hurry to eat deer meat at the moment. Because they won't know if it causes issue in humans sometimes for like a decade down the road. Okay. Questions about that, though? But if you ever see a deer that's acting funny, like walking around in circles, or if it's like has like these like shakes, it probably actually has chronic wasting disease. All right, that is it though. Questions?